Okay, so this is our 7th biomolecule workshop, and the topic is on 1D NMR methods for macromolecule ligand interactions. And in this workshop, we'll cover 3 NMR methods, including STD-NMR, Water-LOGSY, and CPMG. First, let me introduce our biomolecule staff. Deb and I are responsible for the NMR instruments in the chemistry building and in Mary Sue Coleman Hall. Emily is our faculty director, and Anne is the director of operations. Our contact information can be found in this slide, and feel free to contact us if you have any questions about the NMR instruments or in any NMR experiments. And in the past few years, Deb and I have done several biomolecule workshops. Here are the links to those workshops. The videos are all uploaded to our biomolecule website. And feel free to watch any videos that you are interested in. This workshop is currently being recorded. Later on, we'll share it again on our biomolecule website. Webcams are optional for both hosts and participants. And if given opportunity to be on stage, please check if your microphone is muted or not. Also, feel free to use chat to ask questions anytime. And now we'll move on to our topic on 1D NMR methods for macromolecule ligand interactions. So when studying macromolecule ligand interactions, the most important 2 questions you may have are, does the small molecule bind to the macromolecule? And what is the binding site on the macromolecule? NMR is a very powerful technique that can be used to answer those 2 questions. And in NMR, we can utilize NMR parameters to monitor the binding of a small molecule to a macromolecule. Those parameters, for example, include chemical shifts, relaxation rates, NOEs, nuclear over-Hauser effects, and so on. For example, the chemical shifts of a free ligand in solution are different from a bound ligand. Also, the chemical shifts of the macromolecule will change if the ligand binds to the macromolecule. The relaxation rates are different between the free ligand and the bound ligand. The NOEs, which we'll explain NOEs later, the NOEs are also different between the free ligand and the bound ligand. So we can monitor those NMR parameters by using 2 kinds of NMR experiments. One is ligand observed experiments, and the other is macromolecule observed experiments. Here I'm using protein observed experiments as an example. In the ligand observed experiments, we focus on the signals from the ligand. And here's an example. This is a 1D spectrum, and the gray spectrum is from the free ligand, and the blue spectrum is after you add the macromolecule into the ligand. So you can see that the chemical shapes of the peak of the ligand peak changes, and the line shape also change. So based on those spectrum, you can say that this ligand binds to the macromolecule. And in the protein observed method, we focus on the signals from the protein. Here, this example is a 2D proton nitrogen HSQC spectrum of the protein. So this cross peak over here is from the protein signal. So you can monitor the change of the protein signal as you add increasing amount of the ligand. As you can see, those signals are colored by different colors. So the chemical shifts of the cross peak change as you add increasing amount of ligand. And then based on this spectrum, you can make the conclusion that the ligands bind to the protein. Also, if you can assign this cross peak to a specific amino acid in the protein, and then you will know the binding site. So this ligand observed method can be used to answer the question, does the small molecule bind to the macromolecule? And those protein observed method can be used to answer the question, what is the binding site? So in this workshop today, we'll focus on this ligand observed method. The ligand observed method that we'll cover in this workshop includes STD-NMR, Water-LOGSY, and proton relaxation edited NMR, which will focus on this called CPMG method. For all of those methods, they have several advantages. First is that they focus on the signals of the ligand, so there's no limitation on the size of the macromolecule. So the molecular weight of your macromolecule can be like 10 kilodalton, 20 kilodalton, or 300 kilodalton. So there's no size limitation. And the macromolecule can be proteins, lipids, or nucleic acids. And there's no need of processing more information about the receptor because we focus on the signals of the ligand. You don't need to know any information about the macromolecule. Those methods only require small quantities of non-labeled macromolecules, generally like 5 to 10 micromolar. And they are all 1D NMR experiments that are very simple to set up and also rapid to acquire. In less than 1 hour of experiment, you can get a yes or no answer on whether the ligand binds to the macromolecule. And those methods also have limitations because we focus on the signals of the ligand. We will not get direct information on the location of binding. So if you want to know the binding site, you have to use other methods like those protein observed method or other X-ray crystallography methods. And these ligand observed experiments are widely used in fragment-based drug discovery. In fragment-based drug discovery, you start with a fragment library that contains hundreds of small molecules, and the molecular weight of those molecules are or less than 300 Dalton. And then you have a drug target, so you want to identify which fragment binds to this drug target. Then you can go through this fragment screening process, and AMR is one of the methods that can be used for fragment screening. So we use those 1D ligand observed experiments to identify which ligands binds to the drug target. Also, there are other methods available like X-ray, SPR, TSA, and so on. So together with those methods, you can verify those fragment heat and also identify their binding mode. And then by the fragment heat optimization, like fragment growing, linking, merging, and so on. And eventually you can develop a lead compound that can bind to the drug target with very high affinity. So if you want to run those AMR experiments, how to set up those experiments and how to analyze them. So in this workshop, we will cover sample preparation, experimental setup, data processing and analysis, and finally, automation for each of those methods. So we will start with STD-NMR. And STD stands for Saturation Transfer Difference AMR. In STD-NMR, here I'm using protein as an example. So in the AMR tube, you will have your protein sample, and also you have ligand and non-ligands. They all are mixed in one AMR tube. So you want to find which ligand binds to the protein. And then we can first record a off-resonance spectrum. So this off-resonance spectrum is very similar to the 1D proton AMR spectrum of everything in your AMR tube. So we will have 5 to 10 micromolar of protein. So those protein concentration is very low, and then you can barely see the signals from AMR spectra. The ligand and non-ligand concentration are around 200 micromolar. So in this 1D spectrum, you can see the signals from the ligand and non-ligand. And then we can apply a saturation pause. So we selectively saturate the protein resonances. And then this saturation can be transferred from protein to the ligand that binds to the protein. While for non-ligand that does not bind, they do not receive saturation. And this saturation can result in peak intensity reduction. Then so we can record this called on-resonance spectrum. In the on-resonance spectrum, the ligand peak intensity decrease and the non-ligand peak intensity remains the same. And then finally, we can subtract the on-resonance spectrum from the off-resonance spectrum and get the different spectrum. So in the different spectrum, the signals from the non-ligand just disappear, and then what you observe is just the signals from the ligand that binds to the protein. So this saturation transfer process is very similar to heat transfer process. So I'm giving you a graph here to help you better understand this saturation transfer difference AMR experiment. So suppose we have a pod. This pod is like your protein. And then the fish inside the pod is like the ligand that binds to the protein, and the fish outside the pod is like the non-ligand. So at room temperature, they all have the same temperature. And applying the saturation pause is similar to applying a fire to the pod. So now the pod is heated, and then the heat can be transferred from the pod to the fish that's inside the pod. So it's similar to the saturation transfer process from the protein to the ligand. So now the temperature of the fish inside the pot increases, while the temperature of the fish outside the pot, it remains the same. And similarly, you can do a temperature subtraction and get this temperature difference, which is similar to the difference spectrum that we observed in the STD-NMR experiment. And this is how a real STD-NMR spectrum look like. On the top, this is a 1D reference spectrum that contains the signals from your ligand and non-ligand. And then on the bottom is this STD spectrum, or the difference spectrum, where only those ligands that binds to the macromolecule show the signal in the STD spectrum, while the non-ligand signal just disappear. And so to run those STD-NMR experiment, the first thing is to prepare the sample correctly. So we'll start with sample preparation. And there are several considerations, like protein ligand ratio or macromolecule ligand ratio, the buffer conditions, ligand quality control, and negative controls. So let's first look at protein ligand ratio. So I'm using protein as an example, but actually this applies to all kinds of macromolecules. And the ratio depends on the molecular weight of your macromolecule. If you have a 30 kilodalton macromolecule, then you need about 10 micromolar. And if you have a larger size, like 300 kilodalton, then you just need 2 micromolar of protein in the STD-NMR experiment. The ligand concentration is around 200 micromolar. And the ligand concentration does not have to be exactly 200 micromolar. The range is from 100 to 500 micromolar. Also, this depends on the solubility of the ligand in the aqueous buffer. So the ratio generally is 1 to 20 to 1 to 100. And once you have the concentration determined, then you need to pick up the correct buffer to run STD-NMR experiment. The buffer we recommend is phosphate buffer. The example is 10 millimeter potassium phosphate, 50 millimolar sodium chloride at pH 7. We recommend using phosphate buffer because phosphate buffer does not have any hydrogen in it, so it will not give any proton peak in the 1D NMR spectrum. If you are using other buffers like trace, HEPIs, glycerol, or any protonated organic solvents, they may have huge proton peaks that may overlap with your ligand or protein signals. So if you're using those kinds of buffer, make sure you use their deuterated form, and so it can minimize the background signals. And next is about ligand quality control. When running STD-NMR experiment, you will need to first prepare ligand stock. Those ligands can be first dissolved in deuterated DMSO and to a very concentrated concentration, like 10 millimolar to 100 millimolar. And then you will need to test the solubility of your ligand in the buffer you choose. The solubility test can be done by adding small aliquots of your ligand into the buffer you choose, and to reach a final concentration about 200 micromolar. And if the ligand is poorly soluble in the aqueous buffer, as soon as you add the ligand into the NMR tube, you will see precipitates form. If that happens, you can add a more deuterated DMSO to improve solubility. And the maximum deuterated DMSO that is allowed in this AMR sample is 10%. And once that is done, then you can run a 1D proton spectrum of the ligand to check if you can see any ligand peak in the NMR spectrum. Also, you can further quantify the ligand concentration. And if you want to do that, you will need to add BSS or TMSP as a standard reference, so you can quantify the ligand concentration. Here is an example showing you how those 1D NMR spectrum can be used to test the ligand solubility. So on the left, this is a 1D proton spectrum of the ligand dissolved in a buffer that does not contain salt. So you can clearly see those proton peaks from those ligands. And however, if you dissolve the ligand in a buffer that does not that contains low salt, then the ligand just precipitates out, and then in the 1D NMR spectrum, you can barely see the peak from the ligand. So that tells you the ligand is not very soluble in this low salt buffer. And running those 1D NMR spectrum of the ligand sample is also important, as these 1D spectrum can be used to help to check the structural integrity of the ligand. Sometimes those compounds can undergo aggregation or degradation in aqueous buffer. So running those 1D spectrum can confirm that all compounds are what they are thought to be. And this spectrum on the right is an example. So the top is the spectrum of the ligand dissolved in the aqueous buffer. And then the bottom is the spectrum that's taken 4 weeks later. So you can see this sample undergoes degradation. So some peak intensity decreased and some peaks just simply disappeared. And if you see this happens, then you need to prepare a new batch of ligand stock to run the STD-NMR experiment. And finally, it's important to include a negative control sample when running STD-NMR. This is because that those ligands, when they are dissolved in aqueous buffer, they may undergo self-aggregation. So they will form very large aggregates that can behave like a macromolecule. And if they behave like a macromolecule, they will give positive peaks in the STD spectrum. So those aggregates actually is a false positive. And to eliminate those false positive, you can run an STD-NMR spectrum of ligand alone sample. So this here the spectrum on the right shows you the behavior of those non-aggregator and aggregator in solution. So on the left, this is a compound that does not aggregate in solution. So as you increase the concentrations of the compound in the NMR tube and record the 1D NMR spectrum, you can see that the only thing that changes is the peak intensity. The line shape is still sharp and strong, and there's no chemical shift changes. However, for an aggregator, so this compound will aggregate into large aggregates. Then as you increase the concentration of the compound in the aqueous buffer, you can see that some peaks started to get very broad at high concentration. And also there are minor chemical shift changes as you increase the concentration of the compound. So those aggregator, they may get saturated by those saturation pause that we applied to the macromolecule, and then they will give positive signal even without adding this macromolecule. So that's why we need to run those ligand alone sample as a negative control. So here's an example that on the top, this is a STD spectrum of protein together with ligand. And we can clearly see those positive signals, and you may make the conclusion that this ligand binds to the protein. However, if you also run a control experiment with ligand alone, that is without adding the protein, you can see there's still positive signal in the STD spectrum. And then that tells you this ligand may aggregate as solution, so it also gives positive signals at the negative control experiment. And then this ligand should be eliminated for further analysis. All right, and if there's no more question about this STD-NMR sample preparation, then we'll move on to how to set it up on the STD-NMR manually on top spin. And so when setting up an experiment on top spin, the first thing you do is to select the correct parameter set. So this not only applies to STD-NMR experiment, but to any NMR experiment you want to run. So you want to select the parameter set to run those experiments. A parameter set contains all of the information necessary to run your experiment, including pause sequence, acquisition parameters like sweep width, durational pauses, and so on, and also processing parameters. And in TOSPIN, there is a table, so it's under this directory. So that contains hundreds of parameter sets. And those parameter sets can be used to run almost all kinds of AMR experiments. So which parameter set do we choose to run STD-NMR experiment? So to do that, you type RPAR in the command line, and then that will open up this parameter set window. And then you can see there's a list of different kinds of parameter sets available. So in the search line, you can search for STD star. Star just represents any other kinds of letters. So you want to find any parameter sets that can be used to run STD-NMR experiment. So once you search for those parameter sets, and then there will be another table showing up that there are 8 parameter sets available that can be used to run STD-NMR experiment. And among those 8 parameter sets, 6 of them can be used to run 1D STD-NMR experiment. So I've listed those 6 parameter sets here. So all of them can be used to run 1D STD-NMR experiment. And in fact, if you choose any of them, you can get an STD-NMR data. And the one I recommend to use is this called STD-DIFF-ESGP.3. And this one is most widely used for STD-NMR experiment. So we'll explain what this parameter set is doing. Once you load the parameter set, you will see that it uses this lowercase STD-DIFF-ESGP.3. So use this pause sequence to run STD-NMR experiment. So STD-DIFF just stands for STD difference NMR experiment. ES stands for excitation sculpting. So this is a technique that is used to suppress water. So in our NMR sample, there are a huge amount of water, and this can give a very huge water peak in the NMR spectrum. So you can use this excitation sculpting technique to suppress water. GP stands for using gradients. So if you want to know the meaning of those 2 character code in the pause sequence, you can go to this directory here and open up a file called popprogram.info. And if you open that file, you can see there is a list that's showing you the meaning of those 2 character codes that are used for the name of the pause sequence. So if there are some pause sequence that you're not sure what the meaning is, you can check this file over here. And now let's look at this pause sequence in detail. And so in this pause sequence, there are 4 important elements. The first one is that this spoil pause. So we start with the spoil pause that contains 2 short train pause together with a gradient pause. So those train pause and gradient pause, they work like a eraser, and they can remove any unwanted residual magnetization at the beginning of the experiment. So after this step, we are at a good starting point to start the experiment. And then there is a second one. This is a saturation pause that we apply to the protein resonances. This is a shaped saturation pause and can selectively saturate the protein resonances. And then the third important element is this spin lock pause. This spin lock pause is used to remove the broad protein signal. Remember, we still have about 5 to 10 micromolar of protein in your sample, and those protein can give very broad peak in the NMR spectrum. So we can use those spin lock pause to remove those broad background signal. And finally, we have this water suppression element. So that's the excitation sculpting pause sequence we just talked about. So this part is used to remove the water signal in your NMR spectrum. All right, and to run those STD-NMR experiment, you need to define the pause length, pause width, and power level for each of those pause. And fortunately, you don't have to do it by yourself. Instead, you can just use a command called getProsol. So this getProsol command can be used to read all those pause parameters from a Prosol table. So once you type that command, you can load all the pause parameters, and then there are other parameters that you may need to adjust based on your sample conditions. So we'll go through those other important parameters. The first one is D20, which is saturation time. That is how long you apply this saturation pause to your macromolecule. And this pause can range from 0.5 to 5 seconds. By default, it's 2 seconds. The longer the saturation time, the higher the ST signal in your NMR spectrum. And the next important parameter is called D1, relaxation delay. That's the delay between each scans. The D1 needs to be equal or higher than D20. And so by default, it's set to 2 seconds. If you set D20 to like 5 seconds, then D1 also needs to be set to 5 or higher than 5. And D20 is spin lock time. That is the length of this spin lock pause over here that is used to remove the protein signal. And it can be around 10 to 50 milliseconds, and 25 milliseconds is a good starting point. The longer the spin lock time, then the reduction, you will see more reduction of the protein peak in the NMR spectrum. Next, two important parameter is SP9 and constant 62. Those parameters are used to set the power of the shaped path that is used to saturate the protein resonances. If you go to acquisition parameter tab under ZG options, if ZG option is empty, then you will find a parameter called SPW9 that is used to set the power of the shaped path. So you can set it to something around 40 to 60 dB, and 50 dB is a good starting point. And if under ZG options, if DCAC power is already entered into ZG option, then you will need to use this called constant 62 power, constant 62 parameter to set the power of the shaped path. This range of this constant 62 can be 20 to 200 hertz, and also you can lower the constant 62 for lower power, and generally you can just start with 200 hertz. And the next important parameter is this FQ2 list. So you can go to acquisition parameter tab and click this edit button to edit this exam_std file. So in this exam_std file, there are two frequencies that are already entered into the file. One is -40, the other is 0. So this -40 ppm, it's the off resonance frequency. So we apply a saturation pulse at -40 ppm and then record the off resonance spectrum. So this needs to be something that is far away from the protein and ligand frequencies. And this 0 ppm is the on resonance frequency. So this is the frequency that you apply the saturation pulse to saturate the protein. So generally it can be in the range of 0.5 to -1 ppm. So 0 ppm is a good starting point. All right, so that's for experimental setup. And then we'll take a look at data processing. And processing STD-NMR experiment, STD-NMR spectrum is very straightforward. So you can just type this command called STD split in the command line, and this is an AU program that can automatically process your STD spectrum. So it will split your data into two spectrum. One is the reference spectrum or the off-resonance spectrum, and the other is the different spectrum or the STD spectrum. So those spectra are stored in 100, 1000, and 1001 is in the example I given here. And also, when you look at the reference spectrum, you may need to adjust the phase of your spectrum to make sure all the peaks are positive. And once you adjusted the phase, you need to go to processing parameters and check the 0 order and 1st order phase correction value. So make sure these values are the same between the reference spectrum and different spectrum. So that can ensure both spectrum are processed identically. And finally, this is how you present STD-NMR spectrum in a manuscript. On the top, you will have the reference spectra that contains signals from the protein and ligand. So the ligand peak is at higher intensity here. And then you have the different spectrum showing those positive signals, and that indicates the ligand binds to the protein. And finally, you have a ligand alone spectrum. That's the negative control spectrum. And there should be no peak in this difference negative control spectrum. So showing this ligand does not aggregate in solution. And finally, I want to mention that STD-NMR is also a very unique tool that can be used for epitope mapping. That is, it can find which part of your small molecule is closer to the macromolecule upon binding. So this can be done by recording a series of STD-NMR spectrum with increasing saturation time. So that's the D20 saturation time. So it can be increased from 0.5 seconds to 5 seconds. And as you increase the saturation time, the STD signal in the STD spectrum also increase, as you can see here. So we can quantify those signal intensity. The graph on the right shows you increase the saturation time, the signals from the STD signals from each proton in the small molecule also increases. And at a given saturation time, for example, let's go to 2 seconds. So proton 7 shows the highest STD signal. So we can set the relative STD of proton 7 to 100%, and you can put it onto the structure of the small molecule. And this is followed by proton 1, proton 5, and proton 4, and the methyl group. So we can calculate the relative STD accordingly, and now map onto the structure of the small molecule. So now we know that number 7 proton is closest to the macromolecule upon binding. This is followed by number 5 and number 1. And number 4 and the methyl group is distant from the macromolecule. And number 9 and 10, they do not have STD signal. This indicates those 2 protons are more distant from the macromolecule upon binding. All right, so then we will move on to the next method called Water-LOGSY. Water-LOGSY stands for water ligand observed via gradient spectroscopy. And this method is complementary to the STD-NMR method. So in Water-LOGSY, instead of saturating the protein resonances, we saturate the water resonances. Here you can see that we apply a pulse to the water resonances. And since water is continuously in contact with bound ligand and free ligand, it can then transfer this saturation from water to those ligands through a process called NOEs, nuclear over-Hauser effect. So for those ligands that does not bind to this macromolecule, it experiences positive NOEs and it shows positive peaks in the AMR spectra. And for those large molecules, for the ligand that binds to the macromolecule, they experience negative NOEs and they have negative peaks in the AMR spectrum. So here at the bottom, this is showing you that in the Water-LOGSY spectrum, when there's ligand alone sample, all peaks are positive. But after you add the target, adding the macromolecule, those ligands peak is negative and the non-ligand peak becomes positive. And this is a real AMR spectrum of the Water-LOGSY experiment. On the top, this is a 1D proton reference spectrum that contains the positive peaks from the ligand and non-ligand. And at the bottom, this is a Water-LOGSY spectrum. So only this ligand that binds to the macromolecule shows negative peak, and the non-ligand shows positive peaks. Sample preparation of Water-LOGSY is very similar to STD-NMR experiment. The sample that you use for STD-NMR can also be used for Water-LOGSY. So the requirements for the protein and ligand concentrations and ratio are exactly the same. And also the buffer condition is the same, use phosphate buffer. You also have to check the ligand solubility and the structure before running the Water-LOGSY experiment. And finally, aggregation is also a problem in Water-LOGSY experiment. So also you need to run a control experiment to eliminate the presence of aggregation. And also false negatives can be a problem in both Water-LOGSY and STD-NMR. Both Water-LOGSY and STD-NMR, they are best suited to the detection of moderate to weak affinity binders. So KD is in the range of micromolar to millimolar. If you have strong binding ligands whose KD is less than micromolar, then the residence time on the protein will be too long, so they can be a false negative in such case. So if this happens, you will need to use other orthogonal methods like X-ray crystallography or those protein observed method to identify those strong binders. All right, then let's look at the experimental setup. Again, we want to first select the parameter set for Water-LOGSY, and you can search for the Water-LOGSY experiment in the parameter sets list. So there will be 2, and I recommend using this screen_W-LOGSY to run the Water-LOGSY experiment. And this parameter set uses POS program ICONNMR GPNO.2. So let's look at the meaning of those this POS program. And ICONNMR stands for Enhanced Protein Hydration Observed Through Gradient Spectroscopy. So this POS sequence initially is used to study protein hydration. So this POS sequence is named in the way to run Water-LOGSY experiment. And GP stands for using gradients, NO stands for using with nosy mixing sequence. So this is exactly the same mixing sequence that we use for nosy experiment. Then let's look at the pause sequence program. And so we'll first start with a water inversion pause. And the purpose of this pause is very similar to the saturation pause that we use in STD-NMR experiment. So after this water inversion pause, we inverted water resonances, and then that initiate magnetization transfer from water to the ligands and non-ligands and proteins through this mixing time. And this is by a process called nuclear over-Hauser effects. So this mixing time is exactly the same as the mixing time in the NOSI experiment. And then finally, we have this water suppression element. This is also use excitation sculpting to suppress water signal. And the most important parameter for the Water-LOGSY experiment is D8 mixing time. And that's this mixing time here. So the range for this mixing time can be from 0.8 to 2 seconds. By default, this D8 is set to 1.7 seconds. And the higher the mixing time, the larger the Water-LOGSY signal that you can observe. So 1.7 is a good starting point. And then for data processing, these are just a 1D NMR spectrum. So you can just process it first by Fourier transform using the compound EFP. And then you need to phase the spectrum correctly. When phasing the spectrum, you want to make sure the bound small molecule signals are negative. And then you can use the residual DMSO peak in your AMR spectrum as a reference. So this DMSO peak will always be positive. And this is how you present the data. And then at the bottom, this is a 1D proton AMR spectrum or reference spectrum of everything in your AMR tube. And in the middle, this is a negative control that contains only this ligand, so compound alone. You can see that even in the negative control, there are negative peaks. So those negative peaks are from exchangeable protons like NH or OH groups. So they are not from those ligands, binding peaks. And then on the top, this is a Water-LOGSY spectrum with protein. And these negative peaks you can see here and some over here, that indicates the ligand bind to the protein. And also this exchangeable protons remains negative in this Water-LOGSY with protein spectrum. Right, and then let's move on to CPMG. And in the CPMG experiment, we monitor the AMR parameter called T2 relaxation time. For large molecules, like your macromolecule, or for any ligands that bind to the macromolecule, they have short T2 relaxation time. So their AMR signal or free induction decay signal can decay very fast. So you can see this over here, this signal decays very fast. And then after Fourier transform, this results in a broad peak. And for small molecule, that is for non-ligand that does not bind to the macromolecule, they have a long T2 relaxation time. So the FID decays very, very slowly. And after Fourier transform, this results in a very sharp and strong peak. So by looking at the shape of the peak and the intensity of the peak, we can know if the ligand binds to the macromolecule or not. And so in this CPMG experiment, and so you can see this CPMG actually stands for this last name of the 4 scientists that developed this experiment. So you have the ligand-only spectrum. In the ligand-only spectrum, all peaks are positive, and they are all strong and sharp. And after you add the ligand, after you add the target, the ligand that binds to a target, you can see that the peak intensity reduces, and also the peak is getting more broad, while for the non-ligand, the peak remains sharp and strong. And so this is a real CPMG spectrum. On the top is the ligand along spectrum, and on the bottom, the non-ligand peak are all positive and sharp, and the ligand peak is very broad, and the intensity decrease. There are minor difference in sample preparation. So for CPMG, if we compare with STD and Water-LOGSY experiment, it requires more protein amount or more macromolecule amount. So you need to double the concentration of the macromolecule in the MR tube. And ligand concentration can also be around 200 micromolar. And then for the other steps are all the same. So we will not go through the other steps again, and then we'll directly go to experimental setup. Again, you can search for any parameter sets that contain CPMG, and then there are 3 of them show up. The first one, this CPMG ES1D, this CPMG experiment use excitation sculpting for water suppression, while the other 2 use pre-saturation for water suppression. So to make the experiment consistent with the STD-NMR and Water-LOGSY experiment, we'll choose the first one to run the CPMG experiment. And this CPMG parameter set use the pause sequence CPMG underscore ESGP1D. So now again, you know, ES stands for excitation sculpting for water suppression, and 1D is just means it's a 1D experiment. And then we'll look at the pause sequence. So the CPMG pause sequence contains 2 important elements. The first one is a CPMG filter. So it has delay tau, a 180-degree pause, a 2 tau, another 180-degree pause, and a tau. And this CPMG filter is repeated for time n. And then we also have the water suppression pause that contains excitation sculpting to suppress water. So let's look at the CPMG filter. So why do we want to add the CPMG filter in this experiment? This CPMG filter has 2 purposes. First is it is used to remove the broad protein peaks, and this purpose is very similar to the spin lock pause that we use in saturation difference MR experiment. So you can remove the broad background peak from the proteins. And then it also can attenuate the intensities of a bound ligand peaks. For example, if we do not add the CPMG filter and run the experiment, so here this black spectrum is ligand only and red spectrum is after adding the target. You can see that the change in the peak intensity is actually very small. And just by this comparison of those peak intensity, you can't be 100% sure on whether the ligand binds to the macromolecule or not. However, if we add this CPMG filter, it can significantly reduce the intensity of the bound ligand. So now if we compare the intensity changes, there's a huge intensity change after we add in the CPMG filter. So now we can easily correlate the peak intensity reduction with a ligand binding. And when setting up CPMG experiment, there are 2 parameters that are most important. The other parameters you can just using getPro, so to set the values and also read the default values. And then D20, which is a fixed delay between the 180 pause, that's this D20 over here. This you can start with 0.5 milliseconds. And then there's another parameter called D21, that is the total CPMG time. And that time can range from 20 to 400 milliseconds. And this time also vary depending on the molecular weight of your macromolecule. So we recommend using 100 milliseconds at the start. And data processing for CPMG is very straightforward. Just to start with EFP for Fourier transform and APK to automatically to do phase correction. And finally, we will talk a little bit on how to run those experiments automatically using ICONNMR. And Bruker has already developed those parameter sets that can be used to run on ICONNMR. And these parameter sets are called screen STD, screen Water-LOGSY, screen T2, that's the CPMG experiment. And there's another one called screen T1 row. This one is very similar to screen T2 experiment. So in this experiment, instead of using a CPMG filter, it uses a T1 row filter to reduce the signals of the bound ligand. So you can select any of them to run, and I've listed the pause program here. There are some required lists that you need to enter the value, but by default, those values are already put into the parameter set. And what you need to do is just to go to ICONNMR and load those parameter set, click on submit, and then those experiments will automatically get run and data will send to you by email. And finally, you may have the question, do I need to run all those 3 experiments or 4 experiments? And the answer is no. So I recommend start with STD-NMR and Water-LOGSY experiment. And these 2 experiments are complementary. Proteins are rich in hydrophobic residues, so they do well in STD-NMR. And nucleic acids are proton sparse and contain exchangeable protons, so they do well in Water-LOGSY assay. And also STD-NMR and Water-LOGSY require the same sample, so you can use 1 sample to run both experiments. And finally, if you want to further confirm if the ligand bind to the macromolecule, you can add a CPMG experiment as a confirmatory assay. All right, so that's all for all those 3 experiments. So I think we have a question. Does CPMG contain same KD limitation to STD-NMR method? So that is for the KD. So yes, CPMG also have similar KD limitation to STD method, and all of them can be used to detect the binding of very weak signal, weak ligand. But for strong binders, they have the limitation that they may have false negatives. Right.